Our version of this lab was focused on analyzing the effects of consuming alcohol and aspirin together, specifically how the combination of these two compounds affects the decomposition of hydrogen peroxide in the liver. We began by researching the individual effects of ethanol and acetylsalicylic acid, the medical ingredient in aspirin, on the enzyme catalase. Ethanol is in fact a nonspecific competitive inhibitor, meaning that it inhibits all classes of enzymes, and we will explain how later on. Therefore, we hypothesized that there would be a smaller volume of oxygen produced in the test case where we added ethanol. In addition, we found a study which concluded that ASA has inhibiting effects on catalase, and so there should be also less oxygen produced in that test case. Another study explains how in the presence of ethanol, the concentration of ASA will actually increase in the body, so we expected that when both compounds were added, the volume of oxygen produced in the reaction chamber would be less than any other trial. To our procedure focused on keeping the volume of liquid in the reaction chamber constant, so in every test case we used 5 milliliters of H2O2 as a substrate. For the control, we added 5 milliliters of water. For the ethanol test, we added 5 milliliters of a 70% ethanol solution. For the aspirin test, we added 5 milliliters of an ASA solution. And for the last test, we added 2.5 milliliters of both the ethanol and ASA solutions.
We can conclude that our results do in fact agree with our hypothesis. While the magnitude of each trial varied, the trends observed were the same. There was lots of oxygen produced during the control, less produced in the separate ethanol and ASA tests, and the least amount of oxygen produced when ethanol and ASA were combined in the reaction chamber.
We measured each trial over 9 30-second intervals, and there were 3 trials for each setup except the control, which had 4. Control tests 1 and 2 were done on day 1, and tests 3 and 4 were done on day 2. For the other 3 setups, ASA, ethanol, and ASA plus ethanol, test 1 was done on day 1, and tests 2 and 3 were done on day 2. The day 2 trials all released a larger volume of oxygen than their day 1 counterparts, even though the same materials were used and the same procedure was followed. It is possible that the catalase was either oxidized or closer to optimal temperature and pH on the second day and therefore more effective. The first is our control, which was 5 milliliters hydrogen peroxide. There is no inhibitor to this reaction. The third trial actually had released over 10 milliliters of oxygen by the last 90 seconds. We could only measure up to 10, so note the values were estimated by trend and visual observation for the last 3 intervals. The second added ASA. The trend for all trials was still linear, and the volume of oxygen released never totaled more than 3 milliliters.
We found that ASA is an inhibitor of the decomposition of hydrogen peroxide. The study stated that it showed a mixed inhibition mechanism, which essentially means that ASA is an allosteric inhibitor. ASA binds to catalase at a site other than its active site, changing its structure and preventing the substrate, hydrogen peroxide, from binding effectively. This slows catalase's ability to decompose hydrogen peroxide, therefore producing less oxygen.
The third trial added ethanol. It has the same trend. It released more oxygen than the trials with ASA, but less than the trials with only hydrogen peroxide. Therefore, it can be concluded that ethanol is a catalase inhibitor, but is less effective than ASA.
Ethanol is a competitive, nonspecific inhibitor of catalase. At the molecular level, ethanol binds to the active site of catalase and doesn't allow hydrogen peroxide to act effectively as a substrate. Thus, the chemical reaction of 2H2O2 into 2H2O plus O2 happens at a much slower rate and less oxygen is produced.
The fourth trial added both. It is reasonable to assume that the 2 inhibitors together would further reduce oxygen production. This had the lowest volume of oxygen released of all the combinations.
Alcohol has been shown to make aspirin much more potent. As well, some studies show that aspirin inhibits the activity of gastric alcohol dehydrogenase, an enzyme that metabolizes alcohol. As a consequence, the blood concentration of alcohol may be higher. Therefore, the mixture of ASA and alcohol leads to an increase in both of their concentrations. A combination of both further inhibits enzyme activity and therefore further reduces the amount of oxygen produced.
Though the procedure and materials were consistent between trials, there were some sources of error. Because there were no measurements under 1 milliliter, results that landed within that margin of our graduated cylinder had to be estimated. Preventing this would have required a graduated cylinder with more precise measurements. Some air remained in the reaction chamber's rubber tube between trials. This additional air was pushed into the graduated cylinder during the reaction, skewing results. Solving this error would require filling the tube with water before beginning each trial, so that the only oxygen released into the graduated cylinder was that from the current reaction. This experiment is significant in its confirmation of the hypothesis that aspirin consumed alongside alcohol is bad for one's health. It also provides a scientific explanation as to why it is bad, explaining diseases caused by reduced oxygen production in the liver, such as acute liver failure. Extended and combined with other information, this experiment can also be used to confirm the quantities each ethanol and aspirin that are safe to consume in conjunction with each other. This biological limit would most likely occur when the liver ceases to produce any oxygen. An experiment that reveals how much aspirin and ethanol would be required to reach this point would be an important next step. Though this experiment reveals the effects aspirin and ethanol have on the liver, the identical test cases but with kidney would reveal some of the other biological consequences of consuming these substances together. Further tests are required to reach these conclusions, but this lab provided a strong base of knowledge before going forward.